Hello biologists, you are watching this video because I've asked you to redo some questions from your Unit 3 exam. If you can open up your exam, I'm going to coach you through so that you take the best approach possible. I'm not going to give you the answers, but I'm going to help you to annotate, to extract the important information out of the question, and then you're going to have another go. So I'm going to read through question 1 in this video. My suggestion is that firstly you have a glance at the question and you try and associate it with a particular part of the study design. So straight away you can see that this is an experimental design question. We've got test tubes, we've got things being added to the test tubes. We also can see that it's a chemical reaction catalyzed by an enzyme here that ends in A's. So we can get a fair bit of information before we even read the text. Now I'm going to read through it for you, and then we'll look at what I want you to pull out and which parts I want you to annotate and pay close attention to. So the question states, some fruit and vegetables, when cut or damaged, go brown in air. Catechol is a phenolic compound found in plant cells. In the presence of oxygen, the enzyme catechol oxidase converts catechol to the pigment compound benzoquinone, turning the fruit or vegetable brown. Then we can see the reaction here. A student investigated the action of the enzyme catechol oxidase obtained from potato extract. The potato extract was kept on ice until needed and was added to the test tube. The results are shown below in this table. Okay, now let's read this in detail. So firstly, we've got a general knowledge fact: things go brown when exposed to air for some fruits and vegetables such as apples and in this case potato. Catechol is a phenolic compound found in plant cells. Okay, that's just information for you. In the presence of oxygen, the enzyme, now we're labeling the enzyme here, we've got the name for it, the enzyme catechol oxidase. I want you to circle that, write the word enzyme next to it. Always keep in mind that this is the enzyme. What does that tell us about this and this? You know that this here, catechol, is a colourless compound and it is the substrate for the reaction. And here we have the products of the reaction. Lots of people got this wrong, so annotate that. Write down substrate, enzyme, products. Okay, you can see you've got some water here. It converts this into a pigment that makes it go brown. Okay, so this is when you imagine you're doing this experiment. Okay, I want you to notice once you've pulled out some of the key things, and actually before we go that far, it says here a student investigated the action of the enzyme, and it's told you the name of the enzyme again. You know it's the enzyme because it ends in A's. Where did they get this enzyme from? They get it from the potato extract. So what they haven't done in this table is added enzyme. They haven't added catechol oxidase itself. They've added the potato extract as a source of catechol oxidase. So you need to circle this and write enzyme in big letters. So we've got substrate, write that here. Enzyme, write that here. Now here we've got distilled water. Now why are we adding distilled water and why are there different volumes there? That's to make everything up to the same volume. Okay, so in total we've got 6 milliliters of our reaction solution in all of these 3 test tubes. Now, if you imagine you're doing the experiment, which columns of this table are going to be left blank at the beginning? So when you're constructing it and you know what's going into each test tube, what you're going to enter is the variables that you're controlling. So you can enter the independent variable there and any controlled variables that you've got sometimes. The things that you don't know at the beginning of the experiment is the colour change. So that tells you that this here, the colour change, the rate at which it turns brown is your dependent variable. And you will only have that information at the end of the experiment. So what I would do is always, when it's an experimental question, is scribble down IV. What is it that you're changing in this situation? Okay, so you really need to consider what's being changed, what's not being changed, and what you're actually observing. So here your dependent variable is the colour change, and what you're observing is the presence of the potato extract with the catechol. So here we've got just our substrate. in this first test tube. The second test tube, you've just got the enzyme. Now it probably has got a little bit of the substrate in, but it's mostly, that's the source of your enzyme, the potato extract. And this third tube has got both your catechol and your enzyme or potato extract. Okay, so get your head around what's in all of those tubes. Now try and work out what the purpose of those are before we go on to have a look at trying to answer these questions. Okay, so I suggest that you just press pause and you have a think about why we might have run a test tube 1 here. You can have a look at the results and that might give you a clue. What the purpose of a test tube 2 is, and then what exactly test tube 3 is for. Okay, now have a look at the questions. So you need to know immediately what the 3 ingredients are for a good hypothesis. So you always have to list the independent variable somewhere in there. It has to be included, what you're changing. You also need to identify the dependent variable, so what observations, and have a clear direction with that prediction, with that hypothesis. So it's not enough to say that when the independent variable is having an effect, it's going to cause a color change. You need to actually state exactly what will result in the color change, and the color change being your dependent variable. Okay, so here, having a look at the table, making sure that you're describing what you're combining or what you're testing and what the prediction is when you combine certain factors. Then B, this was a really tricky one, but the information is there in front of you, and if you look carefully, you probably know the answer to these questions. You need to use words such as control and experimental tubes. Now, have a look at test tube 1. If you can name it and you can give it a scientific name, then do that. But most importantly, tell me the purpose of test tube 1. Test tube 2, the same thing. Have a look at the things that are included in test tube 2 and tell me what the purpose is. What's the experimenter finding out by running this test tube 2? And then what does test tube 3 tell us? What's the purpose of that one? Okay, press pause and try these questions A and B for me, and then unpause when you've completed those. And let's have a little look at section C. So why is it important to keep the potato extract on ice before adding it to the test tube? So let's find that in the text. Let's circle that. The potato extract was kept on ice until needed and was then added to the test tube. So you know that potato extract is the source of the enzyme. So why are we keeping the enzyme cold? Remembering that it's not pure enzyme, it's enzyme in potato. Okay, so it's likely that we're going to have some catechol in there, some substrate for that enzyme. Why? Think about enzymes, think about temperature, because it's referred to ice. This question is assessing your ability to describe the effects of temperature on enzymes. So think about the purpose of keeping an enzyme on ice. Question D. It says copper ions are needed as a cofactor for the action of catechol oxidase. What is a cofactor? A straight recall question. You need to know what cofactors are. You might struggle with subsequent questions if you don't know that. So you are welcome to look it up if you don't know it when you're retrying this question. Now here it says PTU acts as an inhibitor of catechol oxidase. It inhibits the enzyme as it binds to copper ions, and it's told you that the copper ions are the cofactor. Copper binds to a location separate from the active site. So we know PTU inhibits the enzyme by binding to copper ions, which also bind to an area on the enzyme that is not the active site. Would PTU be a competitive or non-competitive inhibitor? Explain your answer. This is a question or a style of question that pops up over and over again. It is a 2-mark question giving you a 50-50, so it's competitive or non-competitive, and asking you to explain the answer. So you don't get a mark for saying competitive or non-competitive. You only get a mark for your justification. You need to do a whereas or a comparative answer. So tell me which kind of inhibitor binds to a location separate from the active site, and then tell me why it's not the other kind. Okay, so the other kind of inhibitor binds to the active site, and you need to use comparative language, whereas. Okay, that's the classic. So you get one for the correct statement about why you've decided it's competitive or non-competitive, and then why it is not the alternative. I'm going to put the section F into the next video because that's quite a long question. So give those questions a go and then move on to the next one when you're ready.